In our last video, we talked about metabolism and catabolism. Just to remind you, metabolism has two parts to it. It has catabolism, which is breaking things down in order to make energy and precursors. Basically, tearing apart your Legos in order to get energy and get like blocks that you can build things with. And the other part is anabolism. Anabolism is using energy and using your resources to make large, complicated molecules, the sorts of things that your body is built out of. Anabolism is going to be the focus of our topic today. There are two general, broad categories of anabolism. The first is biosynthesis. Biosynthesis is making the building blocks. Even if, you tear apart all of the protein that you eat, and all of the lipids and carbohydrates that you eat, you don't always have enough building blocks to do everything that you need to do, right? Maybe you're not getting enough protein, or maybe even the protein that you are getting doesn't have enough of a particular amino acid. If you are eating a lot of plants, like maybe you're a vegetarian, well, plant and animal lipids actually have different length fatty acids, different types of fatty acids. This is why vegetable oil is oil at room temperature, whereas something like butter or lard is solid. It's because they've got different fatty acid components. So the chances are pretty good that you're going to have to make some of your own components. You're not going to get them all from your diet. You also have to make other things like your neurotransmitters, you have to make your hormones, things like that you don't usually get through your diet, but you still have to make them. That process is biosynthesis. The other part of anabolism is polymerization, which is basically what we've already talked about. Taking your building blocks and using dehydration synthesis to make large macromolecules. And the things that you polymerize are mostly those four things that we've talked about. Large carbohydrates, particularly glycogen, proteins, DNA and RNA, or nucleic acids, and to a lesser extent, fatty acids. You don't make large polymers of fatty acids, but you do put trimers, 3 of them together into triglycerides. Biosynthesis pathways are really, the body has a whole bunch of them, and they get kind of complicated, and if you go on to take a biochemistry course, you're going to cover all of that in great detail. I'm going to give you the bare bones basics of biosynthesis, right? So with biosynthesis, you're making things using what's called a metabolic pathway. What I like to think of as getting from A to B. You start with something, usually something very, very simple, what's called a precursor molecule, and then you modify that precursor molecule one step at a time until you get where you need to go. You can't just change it all at once. It's like, well, really any modification, right? If you wanted to, to put a bunch of updates on your house, like you want to build a second floor and an addition and things like that, you don't just build it all at once. You have to put the frame in first, and then you have to change this, and then you have to change that. And eventually, one step at a time, you make progress to where you're going. And there's a different enzyme for each of those steps. So let's take a look at this. And we're going to use the examples of A and B. So we're starting off with A. Right? This is our precursor molecule. We've got it lying around. It's either something that we get from our diet, or it's something that we have a lot of anyways. But we need to make B. B is the amino acid or whatever that we are lacking and need more of. We're going to have a bunch of logical steps along the way, and each one is going to be performed by a different enzyme. First thing that we're going to do, looking at this A, is we need to do things to make it look more like B. Well, B's got this one straight line here, but it's only got one straight line, so we need to get rid of one of these straight lines here. Boom. So now we're going to have an enzyme come in, and it's going to cut, and then we'll remove a piece. Enzymes can do basically three things. They can cut a piece off, they can add a piece on, or they can change the shape without adding anything. So we just cut a piece off. Now we look at it and we go, okay, closer, but not quite there. This straight line is up and down. This is at a slant, so we need to change the shape. So next we're going to have an enzyme that's usually going to be called an isomerase. Isomerase means an enzyme that changes shape. And it's going to come in and change the shape, like that. Now we go, oh, but look at the B, right? It's got these segments up here and down here, and it's got these curvy bits here. So we're going to need to put both of those on. So we'll have enzymes that are often called synthases, although there's a few other names that they can have. But those are going to add pieces on. So here we go. We're going to attach a piece up top, then a similar piece down below. And now we need to put on these little curvy bits, and we're going to have another synthase come in and attach those curvy bits, hook them on, and it goes away, and boom! We have gone from A to B. There's a different enzyme for each step of this pathway. And note, so we've got each step has a separate enzyme, and sometimes intermediates will themselves be useful things. So on our way of going from A to B, we made E. Maybe we need to use E for something else. Maybe sometimes all we need to make is E, and then we won't do this last step. Or maybe we'll take some of this E and siphon it off. Or we could even potentially take this E or this intermediate here and modify it in order to make something else. For instance, we might need to make F. And then the pathway to get to F is going to be very, very similar up to here. And then we just remove this bottom bit and we get F. Now, obviously, we're talking about enzymes and not letters, but the logic is somewhat the same. I'm not going to go over all of the various biosynthesis pathways, but just realize that everything that your body is making, every small molecule that your body is making, hormones, neurotransmitters, amino acids, special carbohydrates, things like that, they're all made using these metabolic biosynthetic pathways. I'm going to spend the majority of time here talking about polymerization and the standard polymerization pathways that your body uses. In your intro bio class, you will probably go over this in much greater detail, but I'm going to give you the basics right now. And we're going to start off with what's called the central dogma of biology. By the way, this is a term that I hate, and it's a concept that I'm not very fond of either. Mostly, I object to the notion of dogmatic science, but this is the term that you will see in textbooks. And the central dogma of biology is that DNA, which is present in the nucleus, always stays in the nucleus. When you need to use a particular piece of DNA, you copy the DNA into RNA in the nucleus through a process called transcription. Transcription means to make a copy. Back in the days before we had like photocopiers and printers and stuff like that, when you needed to make a copy of a book, they were all handwritten, and you had special people called transcriptionists or scribes that would just basically go through the book and make an exact copy. They didn't even have to know how to read. They were just drawing an exact picture of what the letters were. Is it exactly the same? No, it's never exactly the same. It's going to have like different handwriting and stuff like that, but it's going to be in the same language. All the words are going to be the same. They're going to be in the same language. The punctuation will be the same. That's what DNA and RNA are. They're the same language. They both speak the language of nucleic acids. The RNA contains instructions, usually instructions for how to build a protein. Proteins are not built in the nucleus. They're built by ribosomes in the cytoplasm. So the RNA moves from the nucleus to the cytoplasm, and the ribosome will perform translation on it. Now, when you translate something, you're copying the meaning, but changing the language. And here we're going to change the language from RNA to the language of proteins, right? Nucleic acids and proteins are totally different languages. DNA and RNA bind to each other. They're copyable back and forth. RNA to protein, this requires something that speaks RNA on one side and speaks protein on the other. And the translation machinery, which is ribosomes and a special type of RNA called tRNA or transport RNA, transfer RNA, they speak both languages, and they can take an RNA instruction and turn it into a functional protein. DNA is made of nucleotides. We talked about this a little bit when we were talking about biological macromolecules. And if you'll recall, there are 4 nucleotides used by DNA: A, T, G, and C. RNA uses a U instead of a T, and RNA has an extra hydroxyl group on the sugar, but otherwise is very similar. DNA is made in the form of a double helix. You have 2 linear molecules, 2 polymers, which each have a sequence of bases connected to each other. What's called backbone is made of phosphates and ribose sugars. And its connection just goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, back and forth and back and forth. And then hanging off of this backbone, you will have a sequence of bases that determines the functionality. The one helix will bind to another helix by means of what is called Watson-Crick base pairing. It's called Watson-Crick base pairing because it was originally formulated by 2 scientists named Watson and Crick. They were the discoverers of the double helix, among other people. Watson-Crick base pairing always has a particular set of rules based on how the hydrogen bonds line up. And that is that A, which is a big base called a purine, always bonds to T, or U in RNA, and T is a small base called a pyrimidine. See, the purines have 2 rings, the pyrimidines have only 1. G, or guanine, always bonds to C. Again, a purine and a pyrimidine. So you always have a big base binding to a small base. And the way I usually remember this is A and T are both made of straight lines. And so they bind together. Straight lines bind together. G and C are both made of curvy lines. And so curvy lines go together. This is called complementary base pairing. And what it means is that if you were to separate these two things, you could use any one strand to copy the other strand. So for instance, if I was to take this and take this part and just say, all right, that's going away, right? So let's go, this is gone. And then I needed to rebuild that strand, I would look at it and go, oh, this is an A right here. What goes across from an A? A T does. And so I would put a T here. The next is a G. What goes across from a G? A C. Next is T. What goes across from a T? An A. Next is another G. What goes across from the G? Another C. So if you have one half, you can always build the other half. This is the concept of complementary base pairing. One side is the complement of the other, kind of like the opposite, except they don't actually oppose. When DNA is replicated, that is what actually happens. You take your original double helix strand and you split the helices apart. There's an enzyme called helicase that basically unzips the helix. You then have special proteins called DNA polymerases. A polymerase is a protein that makes a polymer. These are DNA polymerases, so they're making a polymer of DNA. And what they do is they start laying down the opposite base. So if this here was an A, it would put a T across from it. If this next one was a G, it would put a C across from it. And it just goes through and it makes the complementary strand. The other thing, the same thing happens on the other strand. There are some complexities associated with it, and you'll probably get into those in your biology class. But basically what you need to know is that you unzip the DNA into two single strands, and then you polymerize to make a new complementary strand. And at the end of it, you end up with two strands of DNA, each of which will have one old strand and one new strand. So see, the old strand is in blue, and this one here is going to have one blue strand and one new green strand. This one's also going to have one old blue strand and one new green strand. This concept, where an old molecule splits in half, and the new molecules each have one old and one new strand, is called semi-conservative replication. So we would say that DNA replicates semi-conservatively. This process takes energy. And the way that energy is brought in is here. If you take a look, this is the old strand of DNA, and the sequence is going, T, G, A, C, G, T. Here is the new strand being built. Remember, the backbone goes phosphate, sugar, back and forth, and then the base sticks in the middle and forms these specific interactions, like the teeth on a zipper. So this new base coming in, right? So we've got an A here. What goes across from an A? Is it a G No. So this G will come in, and it won't be able to form the proper bases or base pairing, so it will be rejected. When a T comes in, it will stick across from it, and note that this that's coming in is what's called a nucleotide triphosphate. So it's got the base, and then it's got the sugar, and then it's got 3 phosphates. Remember how we said that the phosphate-phosphate bond in ATP was really, really energetic? It stores a lot of energy. Here, when we attach a new base on, we're going to break a phosphate-phosphate bond, releasing these last 2 phosphates. Since that's a very high energy bond, it's very easy to get it to break. And so it provides the energy that will attach this phosphate to that RNA. And then these 2 will float away, as you can see there. Transcription is the process of making RNA from DNA. And since they speak the same language, they use the same types of bases. You just swap out a U for a T. U and T both make the same hydrogen bonds. And you have a different, slightly different sugar molecule. But they both speak Watson-Crick base pairing. The differences between DNA and RNA are U instead of T. You have a ribose instead of a deoxyribose for your sugar. And last, at least inside of cells, RNA is usually single-stranded, whereas DNA is double-stranded. So when we have some DNA and we want to make an RNA copy of it, we start at a particular place called the promoter. And what you do is you get the strands in that space, that promoter space, separate. They unwind a little bit, and you start making RNA inside of this bubble. And it's made by an RNA polymerase, something that makes a polymer of RNA. You unwind the DNA in front of the strand, and you rewind the DNA behind the strand. And so that causes the bubble to move forwards. As the bubble moves forwards, you add RNA bases onto it in a process called elongation. And when the DNA strands come back together at the rear part of the bubble, that just displaces the RNA strand, which falls off to the side. This bubble moves down the strand of DNA until it gets to the ending bit that it's supposed to copy, what's called a terminator. And when it hits the terminator, you stop making new RNA, the RNA falls off, and your DNA rewinds together. So when you make RNA, it's not actually a copy of the whole strand of DNA. That would be unwieldy, because DNA is huge and contains thousands of genes. You only want to make a copy of one gene, one small part of that DNA. So you have this promoter beginning bit and terminator end bit, and that tells you what part of the DNA you're going to be copying. This RNA transcript, which is called mRNA for messenger RNA, can then be exported from the nucleus and taken to the cytoplasm and to a ribosome that will translate it. Translation is the process of converting the language of nucleotides to the language of amino acids. There are three main participants in translation. mRNA, messenger RNA, that's what we just made. That was the RNA copy of a gene. It has a special cap, a protein hat on one side of it that is what the ribosome is going to recognize as the start site. There are transfer RNAs, or what are called tRNAs. These are special RNAs. They don't code for a gene. They're functional RNAs that are basically shuffling agents. What they do is they go out, they get the appropriate amino acid, and then they come back and they put that amino acid in the right place. So they're actually what's going to interpret. They're actually what's going to do the translation. So T stands for transfer RNA, but you can also think of that as standing for translating RNA. These are the molecules that actually speak two languages. On one side of them, they contain what's called an anticodon that binds to the mRNA, specifically, can read a specific 3-base segment of the mRNA. And on the other side, they have a specific amino acid that corresponds to that 3-letter code. For instance, the 3-letter code AUG is a word that when translated means methionine. And methionine is an amino acid. So you would have a tRNA that has the opposite of AUG, which would be UTC, right? Remember, A binds with U in RNA. So it would bind to the AUG sequence at its anticodon. And on the other side, it would contain a methionine, which is the amino acid that codon, that 3-base word, means. Then you have the ribosome. Ribosomes are protein-making machines. They don't do the translating, but they do the polymerization. The ribosome is what's going to bind to the mRNA, bind to the transfer RNA, and then take the amino acid from the transfer RNA and use that amino acid to build the next segment of protein. So it's like a machine. You hand it a component, it's going to then weld that component onto the end of its growing protein. Ribosomes are made out of protein and RNA. And what we've found is that the ribosomal RNA, this is a special type of RNA found only in ribosomes, abbreviated rRNA, as you see down here, that's actually the catalytic part. That's what the enzyme is. So since the ribosome has a catalytic RNA portion, most catalysts, most enzymes that we know of are proteins. This is a special one where the enzyme is made out of RNA, and so it is called a ribozyme, meaning that it is an enzyme made of RNA. Here you can see a tRNA. You'll note that it isn't purely single-stranded or purely double-stranded. It has double-stranded portions, and it has single-stranded portions called loops. At one end, it has that anticodon. So this anticodon is UUA. What's that going to read? It's going to read something that is AAU. On the other end is what's called the amino acid accepting end. This brings in the amino acid that this anticodon or codon corresponds to. And tRNAs look kind of L-shaped. So they've got a straight line going to here, and then another straight line going out there. The ribosome has two subunits, both of which have some RNA and some protein on them. And so here you can see the small subunit of the ribosome, and this is the large subunit of the ribosome. The ribosome binds to the mRNA and reads it like a ticker tape, like basically this big long strip of instructions, and it's going to read it 3 bases at a time. And that 3 base long word is what is called a codon, or sometimes a triplet. And each of these is the tRNA. And there are 3 sites for the tRNA: the A site, the P site, and the E site. New tRNAs come into the A site. Then the P site is where the peptide or protein grows from. And what happens is this whole chunk here will be transferred to the end of that one, making something that is glycergluvalase. And then this will be connected to the end of that. At the same time, the ribosome moves 3 bases forwards, or it pulls the tape 3 bases in. So this tRNA moves from the A site to the P site. This tRNA moves from the P site to the E site. And this tRNA moves from the E site out. E stands for exit. Now the A site is empty, and the next tRNA moves in. And then you just do that over and over and over, right? So you move down, a new tRNA comes in, you transfer the old strand to the new tRNA, and then you move 3 nucleotides or 1 codon down again. The tRNA leaves from the E site, new tRNA comes into the A site, you transfer the growing polypeptide strand from your P site to the A site, and then you move down again. And then you just keep doing that over and over and over again as you grow the protein longer and longer and longer, until you reach a special codon called a stop codon. And the stop codon does not have a tRNA. No tRNA will bind it, so no tRNA moves in, and if the ribosome hits this empty spot for long enough, a special protein called release factor, or RF, will come in and cause the growing strand to release. And then you're done making your protein. This whole process is made possible by something called the genetic code. The genetic code is what your body uses, and specifically what your tRNAs use to interpret a 3-base codon and translate it into a protein or an amino acid. And you can see here that this is an illustration of the genetic code. And the way you interpret this is, you go find let's say we're looking for GUG, right? So we would go down and find the first base on the left side, G, find the second base on the top, so that would be a U. Here's where G and U intersect. And then here are all of our GUs, and then we would find, boom, GUG down here. And then you read what amino acid that corresponds to. The amino acid it corresponds to is valine. Few important things to note here. There's like 64 possible 3-letter combinations, and there's only 20 amino acids. That means that each amino acid might have more than one code that codes for it. This is called a degenerate code. So when we say that the genetic code is degenerate, all that means is that it has synonyms in it. You could have more than one word that means the same thing. So for instance, UUU and UUC both mean the amino acid phenylalanine. All of these right here, these 6 words, all mean leucine. Some of them don't have synonyms. For instance, AUG and UGG only have one word. AUG means methionine, UGG means tryptophan. But most of them have at least 2. Second, it has punctuation, right? If you think of this as a language, a sentence is made of words. Each of these is a 3-letter word, and then you make your protein sentence as a sequence of those words. Well, languages have punctuation. What marks the beginning of a sentence? A capital letter marks the beginning of the sentence. And not only that, but that capitalized first word, it's one of the words of the sentence. And we have the same thing here. What marks the start of a protein is this AUG, or methionine. And so that means that proteins will all, or at least almost all, start with methionine. It is also in sentences what marks the end of a sentence. A period marks the end of a sentence, and the period is not a word of the sentence. So we have basically the same thing here. We have 3 stop codons. These do not code for an amino acid. Instead, they code for stopping the protein. And they say, we're done with this protein. Boom. All right, summary, bullet point list. You need to know for this what anabolism is. You need to know the difference between biosynthesis and polymerization. You need to understand the concept of the metabolic or biosynthetic pathway. And you need to know what DNA synthesis or DNA replication is, what a Watson-Crick base pair is, and what the rules for Watson-Crick base pairing are. You need to know what transcription is, and where it occurs, and what it makes. And you need to know what translation is. You need to know what is involved in translation, the 3 components that we talked about, mRNA, tRNA, and ribosomes, which are made of ribosomal RNA. And you need to know what the function of each of those components is. mRNA is the message that is being translated. tRNA does the translating and brings in the amino acids, and the ribosome organizes everything and actually does the welding, does the chemistry that polymerizes the protein. You need to know what the genetic code is and what it means for the genetic code to be degenerate. Incidentally, a mutation is a change in the DNA. And you can see that if you change the DNA, let's say you changed a CUU codon to a CCU codon, you would change the protein that it makes. You would change a leucine to a proline. That may have no effect on the protein. Maybe that just wasn't a very important base, wasn't a very important amino acid. It may have a small effect on the protein, or it may have a huge effect on the protein. You should know that when we talk about, later on, genetic disorders, and also when we talk about cancer, these diseases are caused primarily by mutations in the genetic code that change or destroy the function of particular proteins.